just starting off with, really, I wanted to give a quick intro of sort of what we're trying to achieve here. We're really looking to introduce a podcast series to inform the scientific community about the types of cutting edge technologies out there to help the current grads and postgraduates with their career advice when moving into industry, and also with like a chance for yourself to introduce your story of how you've got to where you are, what you've achieved, and what your future goals are now moving forward from that as well. So, I mean, starting off with, really, it'd be great to hear a little bit about a brief overview of yourself, your background, sort of your university courses, and what you've studied.
Yeah, of course. And I would like to first applaud that, this podcast, like things that you guys are doing, it's pretty helpful. And I'm sure it will be very helpful for people who are interested in this kind of field. I wish I had that kind of thing when I was growing up, when I was starting out in my career. So I'm really excited to be part of this. So yeah, I'm as you know, my name is Sunny Trivedi. I'm originally from India. And so I did my master's over there, and then I came here for my PhD. That was always clear to me that I wanted to come to States and do PhD. And so, yeah, I came here, started my PhD in University of Maryland, Baltimore County. I stayed there for 5-6 years. I learned quite a bit about science. And yeah, now I'm working in a spatial biology field.
Nice, yeah. And how did you find that transition moving over to the US?
It was it really can depend on the personality. I guess for some people it can be nervous, but for me, I am quite a bit of a risk taker, and I really embrace and love the change. So I was over the moon. I was very excited to move here and start to work alongside with like some of the best minds in the field, right? There is science back in India, but at the same time here, there is cutting edge research. All the technologies are here. And so you get to work on that firsthand. And besides that, it's always exciting for at least me to move to a new country, explore that new culture, meet new people, have new experiences. So it was all sunny and yeah, sunshine and the rainbows. Yes.
No, I'm glad that it's been a good transition. Obviously, you've been there in the US for quite a while now, so fully settled in, and you've had quite a successful career so far. Starting off with, it'd be good to hear a little bit about some of the technologies you've worked with and some of the work experiences that you've had.
For sure. And so how I try to divide my career is it's like, so there are three major biomolecules: DNA, protein, and RNA, right? And so I have always had a fascination to work with all of them and try to understand how they work at the molecular level. And so I didn't have that clarity when I started my research, but when I started with my protein work. So back in India, I started my master's thesis. So that's where I learned quite a bit about proteins, how they work, how they structurally they are related to each other, how they interact with each other, and how like these tiny molecular interactions, they impact on a organism level. So even like a small protein, which you can't even see by a naked eye, right? Interaction of these proteins, then that can shape the whole pathophysiological structure of a human being and how it can impact on a macro level. I think that fascinated me a lot, and so that's where I got into the protein. So I learned all the protein techniques. So that includes how to create the recombinant proteins, mutant proteins, how to express them in the bacterial and mammalian cells, and after expressing them, how to purify them, how to understand their interaction between each other, and how or what kind of impact does that have on a as I said, macro level. And so I learned I have in my career, I've learned more about, I would say, techniques rather than technology, because I feel like techniques are much more important than technology because it keeps you very connected with the science at a molecular level. So you actually understand what's happening in the cells in real time. Whereas technology will make your life easier. So technology can always be like the final output, but if you understand the techniques which goes behind that, I think that helps you a lot. So that's where I focused for throughout my career until I finished my PhD. And now I feel like I have a decent grasp of how things happen at a cellular level. I now am venturing into the technology space where I'm trying to understand that, hey, this is the science. Let's take it merge with any of the machines, robotics, AIs, and try to come up with something which can help us do all of this science that we have been talking about at a very high throughput and a very high speed, a very high plex. And so I'm just trying to implement technology techniques to the technology.
Yeah, no, that sounds good. And have you got anything that you'd be able to show us? I think you mentioned previously about some of the techniques you've done when we've spoken before. Or...
I have a small cool video which I would like. I mean, this is kind of a synopsis of whole of my PhD, I would say. And so I did, while I pull up that video, I'll talk a little bit about it. So I did my PhD in a collective cell migration field. So it's kind of a group of cells which migrates from one place to the other place. And so this is not just a tumbling migration where it just gets flowed, such as like blood cells, right? It's a very coordinated movement, a movement in which cells actually have their own kind of mind. They know, or they decide that, hey, let's all get together, go into this direction. We should move at this speed, and even after getting there, we should do this, that, and that. So it's a very conscious kind of movement on the cell's part. And so we try to understand like the signaling pathways that regulate this collective cell migration. So for that, this is one of the slides which I'm hoping that you guys can see.
Yeah, I can see that, yeah.
Yes. So to understand this collective cell migration, we are using Drosophila fruit flies as our model organism. And so what we would do is we would create this mutant flies. So basically we would create mutants, and we would have that gene incorporated into the flies. And in the next F1, F2 generations, we take these flies, we dissect them, we take out the ovaries. They look something like this. And so each ovary is consisted of different aged ovarioles. So this single, let me place a pointer on, that might help. Okay. So this is one of the ovaries. The whole thing is called ovaries, and this single thing is called ovariole, or you can also call it egg chamber. And this is the oldest egg chamber. This is the youngest egg chamber. So this develops into this, to this, to this, and this. So it goes from stage 1 to 14 in its developmental phases. However, the phenomena that we are trying to understand, that happens between stage 8 to 10. So at stage 7, how does this particular green thing egg chamber looks like? It looks like this, where there are about 15 nerve cells, which are these big cells in between. And there is one oocyte. That's the cell which gets fertilized in future. So that is an oocyte, 15 nerve cells, and all of the 16 cells, they are surrounded by a single layer of follicular epithelium. And at stage 8, what happens is at the anterior end of this egg chamber, some of these regular looking epithelial cells, they convert themselves into a motile cell. So initially they are stationary cells, but then they become motile. And so they kind of become these wheels for this whole migration. And so they take out these 2 red cells, which are polar cells. They make a cluster, and then they detach from the anterior end, and they start their migration towards the oocyte. And that's where they do further function. So we try to understand the factors that are involved in this collective cell migration. And for many of these, we try to do some live imaging, where even after taking the ovaries outside the flies, we can still keep them alive for a few hours, and we try to understand how the cells migrate. And so we try to take this cool, I'd say, live migration of these cells, where, as you can see, these cells are labeled with GFP. So they detach from their anterior end, and then they migrate in between the nerve cells, and they migrate all the way to the oocyte. And we kind of overexpress or mutate some of the proteins and genes to understand how certain signaling pathways involved in this collective cell migration.
Yeah, no, I love the video. I love it.
Okay, cool,
just for some of the people in the audience as well.
Yes.
Just to get a bit of an understanding for them, can you tell them a little about GFP and sort of what that use of that was in the video?
Oh yeah, no, that's a great question. So GFP, as we all know, it's the green fluorescent protein. So it's basically a very small size protein, which can light up very easily under the microscope, and we can easily attach, because it's a very small protein. We can attach that to any of the target protein, And then we can, by looking at the GFP, we are basically looking at the target protein because they are at the same level and they are both attached to each other and they would express at the same level. So we have attached this GFP to one of the proteins called UNPAD. So UNPAD is one of the ligands which is only expressed in the cells which become motile, which are migrating through this whole chamber.
Yeah, and I'm sure that gives it like the specificity, like you're saying, only attaching,
Exactly.
and it then shows that fluorescence on the microscope, which is
For sure.
cool.
And we always have controls where we try to make sure that, hey, it's not expressing anywhere where it's not supposed to be, and that gives a bit more confidence in the specificity that we are looking for.
Yeah, no, it sounds good. And what are some of the applications that you know you've worked with, or that you've researched and kind of understand of how this is used sort of broader, either through this sort of research or through medicine even?
That's a great question. And so I will talk more about some of the applications that I did after that I worked with at Akoya Biosciences, which is after my PhD. But for this part, I think it's mostly understanding the science behind the signaling pathways that are involved. For example, this whole migration is regulated by at least 10 to 15 different signaling pathways that we know of. And so some of them is, let's say, JAK-STAT signaling. So JAK-STAT signaling is one of the signaling pathways which is heavily involved, I would say, in cancer metastasis. So it kind of mimics where let's just for a very layman idea that understand, like, for example, this is a tumor. It detaches from one place in our body and then it goes and migrates to some other place and then it infects the other place. So it kind of mimics how cancer metastasis works at some level. And JAK-STAT, hopscotch, there are FGF pathways, EGFR pathways, there are BMS pathways. So these are the pathways which are all involved in the cancer metastasis, and we can understand metastasis that way. And so that is just one kind of output of the pathophysiological feature that could be it could be involved in inflammation, T cell recovery, and many other signaling and disease pathophysiology.
Yeah, no, a lot of my audience work in sort of sequencing and single cell sequencing, so I'm assuming this is where you can then link that back to genes and understanding some of the oncogenic drivers for those different diseases. Is
Yes.
that right?
You can do that. However, this is mostly we are targeting and looking at the proteins. So there is no like any sequencing involved in this part. However, after this, I started my journey at Akoya Biosciences, where that's where we looked at a lot of single cell RNA molecule detection, which kind of can be read out by either microscopy or the sequencing.
Yeah. No, sounds good. I do love the video as well. I think with this sort of applications then, and sort of you're saying what other ways can it then benefit the wider community?
That's a great question. So this is, I guess, whatever I did during my PhD, that was more about just basic research. The goal is to understand the pathway and not have any like direct translational relationship. However, I took this idea where it's a spatial biology, right? You understand how cells and genes and the proteins, they interact with each other on a spatial plane field. But for example, if I want to take a look at one protein, it would take me about a week to stain them, look at them, and then study this, right? So what if you want to take a look at multiple proteins? There was no like high throughput solution at that time. So that's where I started my journey at Akoya Biosciences. That's where I learned that in Akoya Biosciences, they can detect more than 100 proteins on a single tissue section at the same time. And so basically what you do is you take the slides with tissue section on it, you put all the antibodies on it, and then you put it under the scope and it will image for like a day or day and a half. And then it will directly give like spit out an image which has 100 proteins detected at the same time. So that saves a lot of time and throughput, right? And so that's what they were doing. And so I joined the team to do the exact same thing for RNA. And so then we wanted to detect both protein and RNA on the tissue sections. And then I was given a chance to lead a team where we were trying to have this technology ready where we can detect both protein and RNA on the same tissue section. So to come to your question, to answer your question, so if we can have a technology which can detect 100 protein and 100 RNA on the same tissue section, I think that saves a lot of time in different fields of biology. One of the examples being like clinical studies, right? In clinical studies, a patient comes to you, patient asks that, hey, like I'm facing these kind of conditions. What do you think is going on with this thing? So we can take a tissue section from that person, and then we can study that, hey, let's look at this panel of 100 protein and 100 RNA and see which protein is overexpressing, which RNA is overexpressing or underexpressing. And from that, we can make a very good diagnosis that, hey, this person can be suffering from this kind of situation, and let's have our targeted drug therapy for that person. So it has multiple, I guess, magnitude of detection which can be improved, and we can have a very nice prognosis and diagnosis for cancer, any of the inflammation, any of the immunology related diseases.
Yeah, no, it's good to see it have that real world application rather than just building some technology and making medicines off of that. You're actually then being able to link that to the patient as well on top of that, like you say, in actual clinical
Exactly.
uses.
And it's not just the medicine, right? You can always work with prognosis, where if, for example, I'm sure like your parents are healthy, but for example, like let's say somebody's parents are predisposed to like a certain condition, right? And you may have a fear that, hey, will I have this or not? You can always go to a doctor and then have this whole panel run on your sample and see if there is a predisposition for this particular disease or not. And then you can have or change your lifestyle according to that.
Yeah, no, that's great. And for the image there, you mentioned it goes for about 1.5 days. Is the molecules moving quite slowly? Is that why you're doing it over such a long period of time? And then the final image you kind of speed up to make it look faster? Is that sort of how it works?
That's a great question. I think that it's the limitation of the dyes that you can detect a sample with. For example, like for our visible length, the wavelength, there is only certain number of targets that you can detect at the same time. So you can use one of the dyes which is in the range of 480 nanometer, which is like close to GFP, then 550, which is near the Atto channel, and then a bit of far red. So you can detect maybe, let's say, 5 molecules at the same time, because then you run out of all the wavelength. So what you have to do is you have to deposit these molecules onto the slide. It takes some time to hybridize for them. Then you image them. So it takes some time to image the whole process. And after the imaging is done, you have to wash it off and then introduce the next batch. So in the first batch you detected 5 targets. In the next batch, you again do 5 and 5 and 5. So it's kind of cyclic detection, and through which you can have more than 200 targets detected, whether it's protein or RNA.
So it's more like you're doing a few at a time and then sort of putting those videos almost on top of each other to get a better idea. Is that right?
Exactly. Yeah.
Okay. No, that makes more sense. I've always wondered sort of why did it take so long, and that kind of puts together sort of then putting together an actual image and a recording of what it looks like over several images.
Right. Yeah, and if you were to do this manually, I think it takes a lot of time, because like 5 targets 1 week. But if it's an automation, you can easily cut down that time by 95%, and then it's just all automation. So you can just put the sample on the instrument and then be gone. And then after 2 days, the whole thing is ready for you.
Yeah, and that automation is very expensive. But yeah.
Yes, these instruments, if I have to put like a ballpark number, I would say they do cost somewhere around $300,000-$400,000. So almost like the whole setup is going to be like around $500,000. So not everybody can afford that, but it's like for industrial purposes and for clinical purposes, or for some research institution, it may be possible.
Yeah, I think it makes it difficult for university students as well, because like with sequencing, for example, the sequences are very expensive.
Yes.
So very often they do the sample prep for the sequencing and then they send it off to a core, which is quite challenging, because like you've got a lot of these companies that want people to come in with experience using sequencers and all of these bits of experience that they're kind of pricing them out from doing. I mean, one of the main causes of that is really a lot of sort of the patents that are on the market.
Yes.
There are some big players out there that have patents on some of the larger sequences that make it harder for other smaller startups to reduce that price and really come in as competition. What are your sort of thoughts on Like, do you agree with a lot of these patents, or do you think maybe they should be a bit shorter, or like maybe even changing the rules around them? Yeah, what were your thoughts?
I have like pros and cons for when it comes to patents, I guess, for on both sides. For example, I think the patents right now are close to like 15 to 20 years for a single company when you put it out there, which is quite a long time. So, but I feel like it could be shorter, close to 5 or 10 years, because that gives a company enough time to make back all the money that they have invested in the research, and then also profit from them, and also like be the leader in the market because they have all the sample data to kind of do machine learning from them, right? So that should be good enough time, like 5 to 10 years for a patent to last, not more than that. But also then there is a con to that. For example, if there is a shorter patent, that discourages other competitors to innovate, because if there is a shorter patent, they were like, okay, it's only 10 years. After 10 years, it's open. We can have the exact same thing, and then we can be in the market. However, if the patent is longer, that discourages from other people to rely on them, but also come out with their own technology, because they don't want to be waiting for 20 years. So they would rather come up with their own technology, which will compete with them, and that will innovate and push the scientific boundaries a little bit further. So I think that is just good and bad for both the sides, and we just have to be mindful and in the, I guess, the urgency. If it directly helps or innovates patients' like medical journey, I think that will be very helpful for the society, and then you can have a shorter patent life. So it could be something like which can be dependent on the jury, I would say.
The next sort of thing I wanted to look at and discuss was looking at sort of what is it that you love most about working in sort of science and biotechnology?
I think it has always been pushing the scientific boundaries and having this kind of major impact. So when I was growing up, I always wanted to be, when I was a kid, I always wanted to be a doctor. I was like, okay, hey, how can I have a very good impact on a society? And so being a doctor was what came to my mind when I was 10, 11, 15. However, as I grew, I understand a lot more about the importance of a broader impact. For example, if it's a doctor, it's going to be, hey, you are curing that patient. But if you are a scientist, you can work on these different ideas. And if you can have a breakthrough, let's say drug development or a technology development that can impact not just one or two patients, it can impact the whole community. And having that chance of impacting the broader community in a positive way, it inspires me a lot, and it keeps me going always.
Yeah, no, certainly. That's good. I think with talking about sort of your impact, has there been people that have had a massive impact on yourself? Have you got, is it more colleagues that have happened to have this impact, or would you say it's more having that, a lot of people that I speak to have such a close-knit mentor, and that they sort of talk to their mentor about everything and they help them every way possible? Would you say that's more of an impact? What would you say has been your biggest impact?
Impact on me or my impact on the society?
The impact on you, the impact it's had on your work, how much they've been able to help.
Yes, like if we talk about mentors, I think my PhD mentor, she was one of the best scientific mind that I've ever come across. And it's not just the scientific mind, but the determination that it's kind of infectious, because she works in and out all day, and she always has this smile on her face, as if she enjoys this. Like this is, it's like, I'm from India. I love cricket, right? So it's like her playing cricket all the time. So being in a field where she enjoys this all the time. So she's a mom to two very beautiful kids. She has pets. She was the graduate director at that time, and now she's the dean of the department. She's a wife. She cooks meals. She prefers like how home cooked meals, and even after that, she is giving her A+ game in the research, and she manages the lab, which has multiple students, has to write the grants. And even when she
Wow.
invites her to her house, I would see like a jigsaw puzzles getting formed. She would knit. She would do like some sort of painting. So she, even after having this many responsibilities, she would have time for these kind of things. And I get really, really impressed. And I always call her a superwoman because we always cry about, hey, I don't have enough time to do this, that, enjoy the life. But she does it all and always has a very big smile on her face. So that only happens when you are passionate about what you are doing and it does not tire you.
Yeah, I think, like you said there, passionate is the main thing. It's great to see people like
Exactly.
that as well. I think it's
Right.
we always try and aim to achieve and strive like that as well. I think outside curriculum and activities as well is just so important. More like mental health wise as well. Especially in such a fast paced environment in small biotechs is a lot of hours. There is a lot of work there, and a lot of pressure and stress. But yeah, no, what is really some advice that you'd like to give to sort of other postgrads that were in your position, if they're looking for new opportunities, or maybe even if a lot of people aren't sure if they want to move into industry. A lot of people stay in academia for a long time. What would be
Right.
your advice to them?
Yeah, that's a good question. So I feel like I have sort of two different pathways to this. So first of all is many people feel like going from academics to industry is a taboo for some of them. For example, you are working in an academics lab. Students always feel that their mentors would not like it if they go from academics to industry because they do want to have their own family in the academic fields, be there, be have this prodigy going on. However, this is only, I feel like, is in the minds of students only. Professors do not feel that way. They want their students and candidates to flourish in any way that's possible. So I would say, do not be afraid of going into the industry field from academics. It's always well sought after, well thought after, and then it's always having that translational impact to the society that is well worth it. And the second one is, yeah, having fear of not succeeding or a very fast paced environment of the industry compared to the academics where you are used to taking your own time and diving deep into the science. So I'd say like it's a learning curve. Nobody is expecting for us to excel in the first month or 6 months after you join the industry. If that's the case, then please change your job. But otherwise, just take your time, learn, and adapt to this fast-paced environment, but also at the same time also overlook and not be very nitpicky about science. Just focus on the end goal, and that helps you succeed a lot. And it's much better than academic from my sense because you get to be in touch with a lot of collaborative people, great minds in the field, and it's uh, that you get to be introduced with so many new techniques and fields that you will never be able to do during your PhD, because PhD, or like even after PhD, like when you are doing your postdoc, it's a very niche, very focused field. Whereas in industry, you get to explore so many different varieties and flavors.
Yeah, no, 2 points on that really. The first one that you're talking about, that's what I've experienced from what candidates say to myself is their mentors have been amazing. They've even helped them look for jobs. They've helped them write up their resumes. They've really helped them and pulled out the stops. They've reached out to other people for them that they know might work for other biotech companies. So that on that side, I would definitely collaborate with them. On the second point you were saying as well, I think that the fast pacedness of the biotech space, but the main thing is the direct impact that a lot of people say to me is
Yes.
like having that direct impact of your work, that if you're moving into a product development space, you're going to be developing a product that many companies are going to use for their research, or on the other end, you could be joining a research company and doing pure research that might be to develop drugs, or it might be to develop other sort of research applications as well. So those sort of things are really important as well, and that's one of the other main sort of drivers for people moving. But yeah, no, I've really appreciated you coming on the podcast, Sunny. It's
No,
really
thank you for
appreciate you
having me.
joining me. I'm kind of hoping this can help many other postgrads looking to transition into the industry. And if you are looking to make a move in the biotech companies across the US, or if there anyone out there that's looking to build a team, I hope they can feel free to reach out to me, and we can discuss that in more detail. But yeah, thanks, Sunny, for coming on the podcast.